Thank you. And I will say I feel wedged between quite wonderful speakers and the 12-minute charge for the history of clinical trials in autism feels like a lot to take on. So I want to thank Allison, I want to thank Alicia, I want to thank Paul for inclusion as well as this impossible charge. I'm not going to try to truly get through the full history. I'm going to try to use a few examples to share some key principles that are intended to be complementary to the other speakers. We'll see how we succeed in that. So I'm not going to talk about outcome measures as a major focus, and most of the precision medicine discussion you'll hear from others. I think it's always important to put up a disclosure slide. I will note we've partnered, my group has partnered, many other folks have partnered with a number of companies. That in this country is the way that you bring a potential treatment to market. Unfortunately, none of those have yet yielded something that could come to market. I'll also acknowledge that these are some of the folks I've worked with over the years. I'm not going to call out individuals, and this is a history talk. This is not talking primarily about work that my group has been engaged in. So these are some of the key points that I'm going to try to cover. In each case, unfortunately or fortunately, there will also be a caveat. So behind each of these points, there's also a but or a if only, or what did we learn from something that didn't work quite the way that we might have hoped. And as Paul outlined, that's usually the case. If you put up those percentages for phase 1, 2, 3, and the final number down at the bottom, in autism, you'll find that that's lower, unfortunately, than 12%. And so we just have to acknowledge that, and we have to acknowledge at the same time that we are learning from those experiences so that we don't make the same mistakes repeatedly. So I thought it was important to start, even though we've been talking about the Food and Drug Administration, important to start with some of the data on the behavioral side. And that's critically important because that's something that we know is helpful. I think it's also important to note that none of the behavioral interventions that have been tested would have crossed the threshold for FDA approval. So we have to acknowledge that that's a high threshold, and we can believe that something is effective even if it doesn't meet that threshold. And we have to recognize that and figure out how to work through that. This is data from Jerry Dawson and others showing one of the best examples of a behavioral intervention study. I will note the sample size is still smaller than people would want. Things don't always replicate quite the way people would want. But what I really wanted to highlight here is what's moving is IQ and adaptive behavior. And you see that on the left, that IQ gain is something any of us would be thrilled with, right? That's not a symptom or a trait of autism spectrum disorder, right? On the right, you see adaptive behavior, folks who are maintaining their position in terms of adaptive behavior development in comparison to their peers, and the group that did not receive the behavioral intervention that is very intensive over an extended period of time lose ground in comparison to their peers. Again, very meaningful, but not a symptom or trait of autism. And I think that's important to note that the outcomes that we're able to focus on, what we're able to see as a gain, are things that we care desperately about, but not necessarily things that we think of as symptoms or traits. And that's really the story on the behavioral side. There are improvements that can be seen, but they're much less consistent than if you're looking at function in real-world situations. So popular interventions are the next area. This is something that has been found over and over again in autism spectrum disorder, that there will be a really exciting idea that we'll all want to think is effective. And then it's tested, and unfortunately, in most cases, disappointing. And I'll share a couple of those examples. One is the example of secretin, which some folks remember. I realize now it's less familiar to many. This was a fad treatment. Many, many children injected with secretin in the 1990s. After some case reports, then covered on 2020, Dateline, et cetera. And the story here was that injection with something seemed to lead to improvement in lots of different outcome measures. But it didn't turn out to matter whether you were injected with secretin or whether you were injected with placebo. And this is one of the studies, one of the better studies, where you see, and this is a total score on autism symptoms or traits, where you see a pretty steep decline in the secretin group and an even better decline in the placebo group. Alarming. That shifted people's thinking from a period when you actually could publish open label trials in autism and say, lo and behold, somebody improved on fluoxetine. And that was published in a prominent journal, and lots of kids were then treated with fluoxetine or Prozac. So I'm going to then pivot to a more recent one, oxytocin, where in the 2000s, likely tens of thousands of children received intranasal dosing with oxytocin. Similar media attention, a little less than for secretin because people had been burned already. There's actually a good reason to think about oxytocin in contrast with secretin, where it was really hard to wrap your mind around a biological hypothesis. Good hypotheses for oxytocin. Lots of questions about how you actually would get it into the brain, and lots of questions about whether this is something that we were ready to move forward based on lots of pilot studies, some of which were promising. And then this study that we were a part of used essentially the approach that most kids who were getting it were actually experiencing, which is regular chronic dosing with intranasal oxytocin. And here too, and I realize there are two different colors here, but they're a little hard to see maybe from the back. There was no advantage whatsoever of intranasal oxytocin versus intranasal placebo over an extended period of time. There wasn't as dramatic an improvement with placebo, and I think that's important to note. That may indicate that we're learning some things. It may just be a different outcome measure. So I think this is one of the challenges, and this is one of the things I want to why in the world do you see a placebo effect in a neurodevelopmental disorder? And there are lots of reasons we can think about that. Usually when we think about the placebo effect, we're thinking about the power of belief, and sure, there are lots of things that will improve on placebo. And the more intense the intervention, if you're injecting something, if you're doing a surgery, the more substantial the placebo effect is in other areas. But I think it's important to note that really what placebo is doing is controlling for the nonspecific aspects of treatment. And that actually includes a number of relevant concepts. One is simply regression to the mean. Very few people enroll in a clinical trial when they're doing their absolute best, right? That isn't a natural thing to do. You instead join when you're struggling. And you would expect, just like we all fluctuate over time, that some improvement just happens because you didn't join at a time when things were going well. And I think, very importantly, that fluctuation is superimposed on top of development. And just because somebody has a neurodevelopmental diagnosis does not mean that they aren't continuing to develop. And so if our outcome measures are sensitive to development, which usually they are, you will see improvement, and you have to see better improvement on top of improvement, which is a real challenge that we could spend lots of time talking about. There are a number of other things, like the effects of asking questions. So if you ask a question and you're not sure of the answer, you're going to think about that over the next 2 weeks before you come back and you get asked the same question again. We also tend to be caring when we're conducting a clinical trial, and we say encouraging things. Some of those encouraging things can actually be helpful. Sometimes we give somebody a tip as we're at the doorknob on the way out. You could try this. This seems to go well. That can have an effect. So we need to find ways to mitigate those sorts of effects. One is to always insist on placebo. Paul raised the idea that you could do a natural history study. That's only true if you have a really quite severe course that you're able to say that improvement is something you can reliably believe. I will say oftentimes in behavioral intervention studies, we use a waitlist control. It's like saying, don't change until we administer the treatment. So that is not an adequate control. We need to use truly blinded raters. We need to use longer duration of treatment when possible. That may be part of the story where you see less placebo effect with oxytocin or placebo for oxytocin. And then you have to have, in most cases, equal likelihood of each treatment so that people are less likely to believe, oh, of course I'm on treatment because I have a 75% chance. 50% is a better bet. I'm going to very briefly cover interventions for co-occurring conditions. And I will note, this is by and large where we have data for medications in the context of a diagnosis of autism. And I want to just note that all of the evidence is for things that treat things that are not autism, but in people with a diagnosis of autism, which sounds paradoxical, but that can dramatically improve quality of life. The two medicines that have received approval, risperidone and aripiprazole, are medicines that treat or target irritability and agitation. This includes aggression, self-injury. This includes really one of the clear indications that somebody is distressed. And the medicines then, presumably, are relieving some of that distress that's outwardly expressed. These medicines, unfortunately, come with significant side effects. This is the example of risperidone. It's the first study. I will say subsequent studies have shown more placebo effect, but this was the first one. And you see that most of the improvement happens quite quickly. And substantial improvement happens in most individuals treated with risperidone. Now, fortunately or unfortunately, that's the minority of individuals with a diagnosis of autism. This shouldn't be used for most folks, but it can be very effective. We need more options in this domain, and it isn't the sexiest domain, right? It isn't necessarily the most exciting thing to say that I'm going to relieve distress in this particular area in a subgroup of people with autism. It's not treating their autism, it's relieving distress. But this is an example, and this is work that started with Eric London and Helen Yu, looking, although many others dating back further, looking at a medicine that initially was studied in individuals who had aggression following traumatic brain injury, but has been used over an extended period of time, looking at propranolol, a beta blocker, to see if with less adverse events or side effects it may lead to similar benefit. There are also medicines that have been studied for attention deficit hyperactivity symptoms in the context of autism. We can be successful there, and we can relieve difficulty and allow people's development to proceed. I'm going to go backward there. As Paul noted, biomarkers are a really exciting area to show the potential for target engagement, to show that you're doing something in the brain before you can show that you're doing something with behavior. I think they also may be useful in subtyping groups of individuals. And I'm going to highlight in a different way some of the data that Paul showed you with R-baclofen. I'm not going to focus on R-baclofen at all. There are more data coming out that we anticipate being published in the next year or so. Paul could tell you more about that than I could. Suffice it to say, there is some logic to looking at R-baclofen in autism. This slide has some of that logic on it. I'm not going to spend time talking about it. But this goes into some of the data that Paul showed, but extending beyond it. These are some of the outcome measures that were looked at in a trial of R-baclofen compared to placebo in a group of individuals with autism spectrum disorder. Paul was senior author on this paper and told you about some of the refinements that might be made at the level of outcome measures. But I'm going to highlight the one finding that approached statistical significance in this study. And just to say that the primary outcome, social withdrawal and lethargy, showed no difference. Again, you have a hard time seeing a difference between those two lines. But for clinician ratings of severity, you saw some separation. And that separation seemed to increase over time. This wasn't driven by the majority of individuals. It was driven by a subgroup. You can see there people who showed a significant decrease, a substantial decrease in clinician ratings of severity. About 15% of the overall group of individuals who were treated with R-baclofen compared to placebo. So certainly it's not going to be a general treatment for autism, I don't think. There is some hint already of a cognitive and behavioral subgroup, individuals with more language, higher IQ, and that's being followed up on. There also is some interest in looking at this in relation to particular genetic conditions that increase likelihood of an autism diagnosis. And then I think that there's real reason to try to understand whether there's a biological marker that may allow identification of that subgroup that may respond. Now, we also don't know if that'll hold up. But these two trials, which are now completed and harmonization of data, as I understand it, is underway or maybe at this point finished, but final work has not yet been published, will give us a sense of whether that may prove to be true. And then I'm going to very briefly just highlight, because I know we're going to hear more about precision medicine in a moment, some of what we expect to see moving forward. And don't have an example here from within autism, but we'll note now we have, even if you correct for everything that you could measure across the genome, at least 70 genes that we can believe firmly contribute to likelihood of autism, and probably more than 200 that most of us believe at this point. And that opens the possibility for interventions that are based on genetics, as Paul alluded to. There are now examples of this, thinking about spinal muscular atrophy as one such example, where people have been amazingly able to activate a gene that otherwise would be silent, to show improvement in motor function, to show improvement in outcomes that were previously hard to imagine. And these are now treatments that are being infused into people's spinal fluid regularly at many top hospitals. Exciting to think about, not something that we're thinking about yet in the context of most individuals with autism, but starting to think about in some areas like Angelman syndrome. And this is where I think that we have some challenges to think about, where currently our genetic testing happens after diagnosis, oftentimes way after diagnosis, and isn't done routinely. We are likely to, at some point, get to the place where genetic testing actually happens at birth, where whole genome sequencing could be used as part of a newborn screen, where you'd actually have a genetic diagnosis that precedes an autism diagnosis. We do have examples of that. Tuberous sclerosis oftentimes is diagnosed in utero. So that is something that we have been thinking about, but we don't have a lot of examples. So when you think about it that way, if you had a genetic diagnosis now that precedes an autism diagnosis, you would certainly think about those interventions that we think would be broadly useful. But then there's also the question, in which situation would you actually think about an intervention based on genetics? And I think it's a big question. With the leadership of Wendy Chung, who is leading a large effort to use newborn screening as a research tool to understand whether we can pick up some of these conditions, again, before any cognitive or behavioral symptoms might emerge. We're studying at a Columbia and Cornell Center of Excellence our ability to pick up early changes in development in that population that has early genetic risk already identified, as well as the family's experience receiving that information and how they think about decision making. So I'm going to just stop there and note that you'll hear more about precision medicine ideas in a bit. But to just highlight again, there are lots of things that we have learned, even when things haven't delivered in the way that we initially might have hoped in designing a clinical trial. And as we have more learning, I do anticipate that particularly in the area of subgroups as well as precision medicine, that we'll see significant yield. And then we'll just acknowledge funding sources.